What do our brains tell us about dementia? I'm Denis Moura, and today I'll be presenting a machine learning applied to aging dementia and traumatic brain injury study. Dementia, the progressive loss of memory, is one of the most challenging issues in aging populations. It affects around 15 million people worldwide. Health, social, and economic impacts of the condition makes it an urgent matter of public health. Dementia is a complex, multigenic, or even epigenetic syndrome. Some genetic cases are caused by specific gene variations, while many cases remain with unknown genetic cause. In such complex diseases, intricate complex molecular networks are directly and indirectly affected by causing genes. Gene variation, physical traumas, and epigenetic changes may cause imbalances in these molecular networks, leading to changes in pathways that will eventually turn to a specific pathological phenotype. Instead of searching for gene sequence variations, we can look for gene expression profiles in the brain and understand the differences between healthy and dementia samples. The Aging Dementia and Traumatic Brain Injury Study is a database from the Allen Institute for Brain Science. It comprises RNA sequencing data from brains of aged individuals that had a type of dementia or a healthy brain. Starting from the hypothesis that brains with dementia have a differential transcriptome profile compared to control brains, our objective was to create a model that correctly identifies dementia samples based on several genes expression profiles in the brain. RNA-seq data from diseased human brains with and without dementia were collected through the Aging Dementia and Traumatic Brain Injury Study. Our language was used to clean and manipulate the datasets. We assumed the conditions to be dementia and no dementia for the samples, and the analyzed brain regions were temporal cortex, parietal cortex, hippocampus, and forebrain white matter. Our library DE-seq2 was used to normalize and analyze the RNA-seq data. Transcriptome patterns of significantly differentially expressed genes were used as input for machine learning algorithms, decision tree, and naive Bayes. Importance analysis was performed to find the most important genes in the decision tree classification of dementia brains. Lastly, the top 10 most important classifier genes were used to create a random forest model to classify the sample. We found differential transcriptome patterns in dementia brains, with a total of 1,629 differentially expressed genes, 1,550 upregulated and 79 downregulated. Our classification model, based on naive Bayes using all differentially expressed genes, was able to correctly predict over 72% of dementia cases in our test samples. The top 10 most important genes were identified for each brain region. Using the top 10 genes as input, we were able to increase the random forest model prediction potential to around 82%. Comparing the transcriptome patterns in the hippocampus proved to be more efficient in classifying the samples as dementia or no dementia. In conclusion, we have successfully found differential transcriptome patterns for human brains with dementia and identified the most important genes for predicting the condition using machine learning algorithms. Thank you for watching.